Okay, in this lecture we talked about kind of just basic A and P of the nervous system. So the nervous system is broken into two main components. We have the central nervous system, which is comprised of the brain and spinal cord, and we have the peripheral nervous system, which includes the somatic nervous system, which is going to control voluntary movements like skeletal muscle, and also the autonomic nervous system, which controls the automatic functions, things we don't consciously control. And there are two divisions here. We have the sympathetic or the fight or flight division, and the parasympathetic or the rest and digest division. So some terminology here. The brain, of course, is the main integrative organ of the nervous system. The spinal cord is the other central nervous system structure that travels from the brainstem down the vertebral canal. A nerve is a bundle of axons that are found in the peripheral nervous system. A tract, these are bundles of axons found in the central nervous system. A cranial nerve, this is one of the 12 pairs that arise from the brain and brainstem. These are structures of the peripheral nervous system. Spinal nerves, these are peripheral nerves that come from the spinal cord. Ganglia, these are collections of cell bodies of neurons that are found in the peripheral nervous system, and then nuclei are bundles of these neuron cell bodies found in the central nervous system. So the nervous system has three main functions. It has a sensory function, an integrative function, and a motor function. So sensory, this is when the nervous system detects changes, in either the internal or the external environment. And then the integrative function, this is when the nervous system decides what to do with that sensory information. And then the motor function, this is the response after this integration. So once this information is integrated, the nervous system is going to send out some response through motor pathways. So some histology. There are two main cell types in the nervous system. We have neurons, which are the neuroconductive cells of the nervous system. We have neuroglia, these are the supportive cells of the nervous system, and we're going to talk about specific neuroglia in a little bit. So we also have afferent, efferent, and interneurons. Afferent neurons, these are sensory, so they are going to bring information into the central nervous system. Efferent, these are motor neurons, they carry information away from the central nervous system. And then interneurons, these are basically just connections between afferent and efferent neurons. So neurons have three main parts. They have the cell body, also known as the soma or the perikaryon, and this is where you're going to find things like organelles as well as the nucleus. Axons, these are processes that conduct impulses mostly away from the cell body, but they can also bring information to the cell body. And then dendrites, these are kind of sensory structures that are going to sense incoming stimuli. So here is just a little cartoon of a neuron. Here is the cell body or the soma. This is the axon. There's only one axon per neuron, and these are the dendrites. So basically the dendrites are going to bring information in. It's going to be integrated here, and then it's going to be sent out the axon. Information can also come back up this way. And these axons at the end, they kind of branch out, and these branches are known as axon terminals. Myelin. So myelin is a fatty, lipid-rich substance that is found around axons, and they form what's known as the myelin sheath. And the job of myelin is to insulate these axons and increase the speed of conductions down the axon. And these impulses are going to move by what's known as saltatory conduction, where they basically jump from node to node, which are the spaces that are not covered in myelin. These nodes are known as the nodes of Ranvier. Now, white matter, when you hear about white matter and gray matter, white matter is nervous tissue that consists of myelin, myelinated axons. And then gray matter, this is tissue that is unmyelinated, things like certain axons, but also mostly like cell bodies. So here you can see a neuron again. These are the little the bundles of myelin formed by Schwann cells, and these impulses are going to jump from node of Ranvier to node of Ranvier. So it speeds up the impulse down the axon. So the neuroglia or the nerve glue of the central nervous system, we're going to talk about neuroglia of the central nervous system and then the peripheral nervous system. So starting with the central nervous system, astrocytes. Astrocytes are very, very large cells, definitely the most numerous of the cells in the central nervous system, and they're basically going to ensure stability of the CNS. And they do this by a number of functions. Just know that they work to ensure stability of the central nervous system. Oligodendrocytes, these are neuroglial cells that myelinate axons in the central nervous system. Microglia, these are, they're actually phagocytes, they're immune cells, and they arise from monocytes. And then finally the ependymal cells, these are epithelial cells that are going to be involved with producing cerebrospinal fluid. So here we can see some of these cells. Here's an astrocyte here. This is an oligodendrocyte, which myelinates these axons in the central nervous system. These are the ependymal cells, which are involved in the production of cerebrospinal fluid, and these are the microglial cells, which are immune cells. Now, there are fewer neuroglia in the peripheral nervous system, but one of them is a Schwann cell. Schwann cells are going to myelinate axons in the peripheral nervous system, and what they do is they kind of like they do like this jelly roll thing. These are the cells, these are the Schwann cells, and they just lay down these layers in the circular pattern around these axons. And then finally, satellite cells, they just ensure stability of the peripheral nervous system, kind of like astrocytes. Okay, then we talked about action potentials, and this is the way that information travels down axons. And these action potentials, they're all or none events, and they occur by changes in the electrical charge across the membrane. And this occurs by changing permeability to ions across that membrane. Now remember, at rest, the membrane has potential. Okay, it's polar. And the inside of the cell is negative compared to the outside. And the value is about -70, -80 millivolts. You don't need to know the number, but and this negative value is mostly due to proteins inside the cell. Now remember, at rest, sodium levels are high outside the cell, potassium levels are high inside the cell. And that's important because we're going to change those levels in order to move information down this axon. Now, this we change these levels by altering permeability across the membrane. And this is done by opening sodium and potassium channels. Okay, they're called voltage-gated, meaning that they open in response to changes in voltage. But in order to get a change in voltage, we need a stimulus. And the stimulus can basically, it could be anything. So this is any change in the internal or external environment that can alter that resting membrane potential. Now, if that potential becomes positive enough, we will reach what's known as threshold, and that's about -55 millivolts. And once you hit threshold, an action potential will occur. Okay, if you don't reach threshold, it won't occur. Remember, these are all or none events. So once threshold is reached, I'm just going to go to this picture here. So we can see this is the cell at rest, okay, around -70 or so. And then up here, somewhere around here, this is going to be threshold. Once we hit threshold, both sodium and potassium gates are going to open. Okay, so at this point, both sodium and potassium gates are open. And the thing is, the sodium gates are open all the way, and the potassium gates are kind of open only halfway. So we have sodium coming into the cell more quickly than potassium's leaving. And what happens is the inside of the cell becomes positive. We call that depolarization. That's the first step, depolarization. And we actually become more positive than zero, and that's known as the overshoot. Now, once we get to about here, those sodium gates are going to close, so we no longer have sodium coming into the cell, but the potassium gates are completely open. So we have positive charge that's leaving the cell. And if you have positive charge leaving the cell, it's going to start becoming negative again. And we call this repolarization, when we're becoming negative again. Now, eventually the potassium gates will close, and the sodium-potassium pump will work to reestablish the correct concentrations. Therefore, if there's another stimulus, another action potential can occur. Now, there are some other phases in here. The big ones I want you to understand are depolarization and repolarization. So we talked about action potential. Now we're going to talk about how we can take that action potential, that electrical energy, and convert it into chemical energy, so we can communicate between neurons. And again, I'm just going to go to the picture. You can read through the words, but I'm just going to use this picture. Okay. just adjust my pen size. So as the action potential comes down the axon, it gets to a point. Okay, this is called the end bulb, or the synaptic end bulb. This in here is the synapse, or the synaptic cleft. This is the presynaptic cell up here. This is the postsynaptic cell. Okay, and we're going to try to get information to go from this cell to this cell. That's the whole goal. So as we come down the axon here, the action potential moves down the axon. It gets to a point, and it causes the opening of the voltage-gated calcium channel. Calcium is going to come into this presynaptic cell, and calcium is going to be the trigger for the release of these neurotransmitters inside of these vesicles. Okay. So calcium is the trigger to release or exocytose these neurotransmitters into the synapse. Now what happens is, I'm going to draw in some little receptors. These neurotransmitters will bind to these receptors. And when they bind to these receptors change shape and they become like a little channel, like a little pore. And when they do that, positively charged ions, cations, are going to enter into this postsynaptic cell. And that's going to be the stimulus that allows the action potential to move from this cell to this cell. Okay, so this is how the action potential eventually allows communication between one cell and another. Now, once that neurotransmitter is released, most of it's going to bind here on the postsynaptic cell. The rest of it is either going to be broken down by enzymes in the synapse, or it's going to come back and bind to a receptor on the presynaptic cell and be taken back up into the cell. That's called reuptake. So those are kind of the three fates of the neurotransmitters once they go into the synapse. So we talked about a few of the neurotransmitters. So acetylcholine, this is by far the most well-studied neurotransmitter. You see it pretty much in all parts of the nervous system, especially the peripheral nervous system, but also the central nervous system. This is the one that operates at the neuromuscular junction. Glutamate is an excitatory neurotransmitter in the central nervous system, so both the brain and the spinal cord. GABA, which stands for gamma-aminobutyric acid, is an inhibitory neurotransmitter, especially in the brain. And glycine is an inhibitory neurotransmitter in the spinal cord. And then serotonin, again, very important neurotransmitter in the central nervous system, especially the brain, and it's associated with modulation of pain, but also activation of certain parts of the brain for arousal, but also mood and all of those things. The catecholamines, so these include epinephrine, norepinephrine, and dopamine. Dopamine is associated with reward, and therefore it's associated with addiction. Also emotion and the control of movement, especially smoothing out movements. We'll talk about that more when we talk about Parkinson's disease. We talked about epinephrine, which is important in the sympathetic division of the autonomic nervous system, and it's important for fight or flight responses. And then norepinephrine, again, another important neurotransmitter in the autonomic nervous system, and it's associated with emotion and alertness and lots of other things. So then we started to get into just a little bit of anatomy of like the brain and the spinal cord. So the brain, of course, large organ in the cranial cavity. It has four major parts. The cerebrum, so the cerebrum is kind of what everybody thinks about when they think of the brain, but also the diencephalon, which has things like the thalamus and the hypothalamus. The cerebellum, which is an important motor portion of the brain, and then the brainstem, which controls a lot of our vital functions. So the brain and the spinal cord, the central nervous system, these structures are covered by meninges, and these are protective layers that actually have a few other functions as well. The outermost layer is called the dura mater. Okay, dura mater means tough mother. It's a thick layer, and it's very easy to see. The middle layer is called the arachnoid mater, and the innermost layer is the pia mater, and this is directly on the brain and spinal cord tissue itself. And there are spaces between these meninges, okay, these meningeal spaces. So the area between the periosteum of the skull and the dura mater is called the epidural space, and it's a potential space, meaning that it's not really there unless something opens it up, something like a bleed. Now, the area between the dura mater and the arachnoid mater is the subdural space. That's also a potential space. Again, there's really no space between them until something opens them up, like a bleed, like a subdural hematoma. The area between the arachnoid mater and pia mater is called the subarachnoid space and is filled with cerebrospinal fluid. So this is a true space. So here you can see some of these spaces. So here's a subarachnoid space. It's filled with cerebrospinal fluid. This is actually how cerebrospinal fluid will get back into the bloodstream. It'll go through these structures known as arachnoid granulations into the superior sagittal sinus. The blood-brain barrier, so this is a specialized system that limits the fluid environment around the brain, and it's pretty much composed of capillaries that have very, very tight junctions between those endothelial cells. And these tight junctions prevent things from getting into the brain, okay, like pathogens. There are other structures around these capillaries, like astrocytes. Astrocytes kind of wrap themselves around these capillaries to provide just more protection. But of course, some things can easily go through the blood-brain barrier, like oxygen, CO2, alcohol, anesthetics. And it's also deficient in areas of the hypothalamus, the pituitary gland, and the pineal gland because they are endocrine organs and they need to get their secretions into the blood. So you don't want really tight junctions there. You want to allow these hormones to get into the blood. So that's why it's deficient in those areas. Here you can just see some schematics of the blood-brain barrier. So here's the capillary. You can see all of these structures that kind of surround themselves around the capillary. There are also tight junctions between these endothelial cells, so a lot of protection. Of course, things still, in circumstances can get through there. So cerebrospinal fluid, this is a clear, colorless, ultrafiltrated blood. Basically, it's very, very dilute plasma, and it's found in the ventricles, the central canal, as well as the subarachnoid space, and its job is basically to circulate glucose and ions, lymphocytes, oxygen, all of those things. It's produced by these choroid plexi, which consist of those ependymal cells that we talked about earlier. There are 4 ventricles in the brain. Each of them have a choroid plexus. We have 2 lateral ventricles, a 3rd ventricle, and a 4th ventricle. So I'm just going to go to the picture here. The lateral ventricles are connected to the 3rd ventricle by what's known as the interventricular foramen. And then the 3rd ventricle is connected to the 4th ventricle by what's known as the cerebral aqueduct, or the aqueduct of the midbrain, sometimes called the aqueduct of Sylvius. And then the 4th ventricle is continuous with the central canal of the spinal cord. And again, this is that picture showing the subarachnoid space. This is actually how cerebrospinal fluid is returned to the blood. So it goes through these arachnoid granulations, sometimes called arachnoid villi, and it empties into the superior sagittal sinus, which will eventually get back to the venous system. So the brainstem, this is found between the diencephalon and the spinal cord, has 3 major parts, has the midbrain, has the pons, has the medulla. So there are a lot of smaller structures in these 3, but the big takeaway, midbrain, we didn't talk about in much detail. The pons, I talked about the pneumotaxic center, which inhibits excessive inhalation. The medulla controls things like respiratory rate and heart rate and very basic sort of functions. So the hypothalamus, very, very important structure. Structure of the diencephalon, it's inferior to the thalamus. And some of these functions you need to remember. It's basically the boss of the autonomic nervous system, so it's going to regulate the autonomic nervous system. It's going to produce hormones that will go to the pituitary gland. We'll talk about that relationship in a couple weeks. It's also involved in eating and drinking behaviors. It regulates body temperature. It does some other things, but those are kind of the big 4 that I want you to focus on. So here's the hypothalamus. Again, we're going to talk in detail about its relationship with the pituitary in a couple weeks, so don't worry too much about that now. So the cerebellum is a very large brain structure. Posterior to the pons and medulla, it's inferior to the cerebrum. Has gray matter, which is called the cerebellar cortex, and also internal white matter known as the arbor vitae. It's involved with things like balance of motor movements, coordination of motor movements, timing and precision of movements, and any sort of lesion or damage can result in ataxia, which is a lack of that coordination. So this is the cerebellum back here. Again, it's a very large structure. Okay, then the cerebrum. The cerebrum's the largest part of the brain. It has cerebral cortex, which is gray matter, and an inner cerebral white matter. So remember, the brain, actually, the outside is gray matter, the inside is white matter, and then it's the opposite for the spinal cord. So in the spinal cord, the outside is white matter, the inside is gray matter. So these folds in the cerebrum are known as gyri. The singular form is gyrus, and then the shallow grooves are called sulci. Again, the singular form is sulcus. And the cerebrum has 5 lobes. Some people say 6. But it has the frontal lobe, the parietal lobe, the temporal lobe, occipital lobe, and then if you kind of pull the temporal and parietal lobes back, there's another lobe under here known as the insula or the insular cortex. And this I took from your book, just describing some of these parts. Now the spinal cord, again, has white matter and gray matter. The difference is the white matter is on the outside, the gray matter is on the inside. And the gray matter contains anterior, posterior, and lateral horns. These anterior horns, or ventral horns, which are here, they're going to contain nuclei for motor movements. So motor movements are going to originate from this area. Well, they're first going to come from the brain, but they're eventually going to exit the spinal cord through these ventral horns. The dorsal horns, or the posterior horns, are going to receive information. Okay, so information is going to come in through the dorsal horns. There's probably going to be an interneuron between them, and then it's going to go out this way. Okay, so this is afferent information, and this is efferent information. Okay, and again, showing it's kind of the same thing. So this is the dorsal gray horn. Information's coming in, sensory afferent information. It's probably going to be an interneuron or two between these, and then this is the efferent information coming out the ventral horn. And I'm going to stop the video here. I have another video on cranial nerves. I will attach that in the email, and then I will finish the rest of this when we finish it in class. As always,